read the manual, which I recommend you do, just take it. It's super short, as you've seen. It's not long at all. Doing a quick read is great. Solubility basically means being able to dissolve in a substance. And like we know, we saw this in lecture. The big idea when we're talking about dissolving has to do with like dissolving like, right? So if the idea is that like dissolves like, if I am a polar solvent, I'm a liquid that's polar, what substance is going to dissolve in me? So if I'm a polar solvent, the only thing that can dissolve is another polar substance, or, of course, an ionic substance, right? Because polar means a partially charged. So, so yes, it's totally fine as long as you have a like dissolving like. So what if my solvent is nonpolar? What can dissolve in my nonpolar solvent? Because I can have a nonpolar solvent. It just has to be in like relationship for solubility or for dissolving. So if I'm nonpolar, what do I need to dissolve? What is my solute that will dissolve? Another nonpolar. Exactly. Right? Exactly. So anything that's fat-based, that's nonpolar as well. It's the same idea, is going to be able to dissolve there. Most of the time, when we're talking about solvents or the solution that something dissolves in, we're usually referencing water. Like that's kind of the universal idea here, but it's not, we can have anything dissolving in anything else as long as it's like dissolving like. Okay, guys, so that's what we're looking at here. So again, we're going to review the idea of polar and nonpolar, and we talked about that in lecture, and you guys did a great job. So we're just going to see how this actually works in the lab. You get in groups, you talk about it, blah blah blah. You guys will just talk about it together with me. So that's it. All right, awesome. So let's go back to my PowerPoint, and I'll jump back and forth, whichever one kind of suits my needs at any given time to give you guys proper instruction. All right, so this is just a complete review for you guys, right? The idea that if you're going to dissolve in something else, right, you're soluble, you have to have that like dissolves like relate, right? And so that's what it says here. So some of the stuff that's super redundant, because I've already gone over it in lecture, I'm not going to like belabor points. When I first introduce it, I repeat and repeat and repeat. Here we're just going to bring it up because we already know what this is, presumably. If you don't, please review it. This is on your test next week. If you have any questions, please come onto the discussion board. You definitely want to make sure that you're getting any questions answered. So the discussion board is great. So go get on there for lecture. All right, so again, ions is covalent bond. When it comes to covalent bonds, a polar covalent bond is going to be able to interact positively with an ionic bond because that polar covalent is going to be charged. And so that's why we see like dissolves like. These guys are not like. They're not going to be happy together. Okay. The idea here is that you have electrons. Happy molecules have 8 electrons, except for hydrogen. They're like these little weird things, and they're happy with 2. And then again, the idea of just describing an ionic bond where you give an electron or you take an electron, and that satisfies each of the atoms' need for 8 electrons. Just to review. All right, and then we have this, which is an ionic bond. I donate an electron, I become positive. I accept an electron, I become negative. And now I can interact by attracting one another with a negative positive charge. All right, yay, that's it. So what I want you to do then is look on page 15, and the information that you see on this slide is related to the information that you see on page 15. Okay, so we're going to start these experiments. So the first thing that you need to know is that limonene, or limonene, but I'm going to go with limonene. Limonene is a non-polar solvent, right? So you've got to know right off the bat, okay, what's going to dissolve in this, right? You know the only thing that's going to dissolve with limonene is something that's non-polar. Why? Like dissolves like, okay? And then water, well, you know water is a polar covalent. Wow. I just put solvent and covalent together in one word. I'm famous for that. But that's not a real word. Polar solvent means it is going to be the liquid that is going to have our solute dissolve in it. Okay, so solvent, in case you're, no problem, Cheryl. Oh, okay. So the solvent is going to be usually a liquid, that the solute, which is usually a particle-like sodium chloride, salt, is going to dissolve in. So that's where solvent comes into. Okay, so we have a non-polar solvent, which is limonene, and we have a polar solvent, which is water. We already know what polar is, right? We know that it's partially charged. So you have to have some sort of charge to be able to dissolve in water. Okay, so we know that. And then again, just as a reminder, here's the definition of solvent: something that dissolves other substances, right? In other words, it's going to be the water that you pour your sodium chloride in, or sugar, any solute that's charged, and it will dissolve, or it will be soluble. Okay, so that's kind of the idea there. So if you flip over to page 16, again, everything that's happening is happening on the slide, so it gives you enough information, but so that you can kind of follow along and take notes if you'd like to do that. Again, this is content that we're going to see on the lecture. Unless it's on your getting ready for the lab midterm, we're not going to see this on the lab exam. All right, so here is my reproduction of the chart on page 16 in your lab manual. And what we have here is an unknown substance. We don't know what that is. We don't know if it's polar. We don't know if it's non-polar, hence the idea of being unknown. Okay, what we do know is that we can experiment and see, well, what happens if I put this unknown substance in a water solvent? In other words, in a polar solvent, what's going to happen? And then likewise, well, what if I put the same unknown substance into a limonene solvent or a non-polar solvent, right? And then we're going to see what happens. By that, we can deduce, well, what type of substance is this? Is it a polar substance or is it a non-polar? I can't tell you what it is because I have to do actually some it's not microscopy. Oh, I can't remember. It's basically a chemical test where you can break down all the atoms in it. And I'll remember it in like 10 minutes. We don't do that, obviously, in anatomy and physiology. That's something you do in organic chemistry. Here, we just want to know, hey, is this thing polar or non-polar? So let's take a look at what we can do, what we can deduce from what happens in our experiment. All right, guys, so if I put my unknown substance into my water, which is solvent, and it dissolves, what does that tell you right off the bat about this unknown substance? What is it? It's polar, right? How do you know? Well, because we know water is a polar substance and like dissolves like. Boom, we're done. We just figured that out. But we're going to keep going because we can make a couple more deductions from this, right? We can deduce some more ideas. Now, we're going to confirm this by putting this unknown substance into limonene, which is nonpolar. Now, if this substance is polar, will it dissolve in a nonpolar solvent? Mm-hmm. So can I put a polar substance in nonpolar? No, I can't. It's not going to dissolve. It's going to basically hang out on the top if it's denser, if it's heavier. That's it. Exactly. It's not soluble. Boom. We've confirmed this. Now, we're going to we're going to take some of this deduction skills that we've just learned, and we're going to apply it to the body, right? Because we know that the body has certain characteristics, right? For example, we know that the blood is primarily what? What is the blood primarily made of? It's primarily made of water. And guess what? Water is what? Polar or non-polar? Water is going to be polar. Awesome. Sweet, guys, right? So that means that I can deduce from the information that you just told me, the unknown substance, will it be soluble in blood? Yes or no? Yes. Yes, it will be, because we know that the unknown substance is polar. We know that blood is predominantly made of water. We know that water is polar. Wow. I am on a roll today, guys. So you're going to have to translate some of my English. So the unknown substance then is going to dissolve in blood because the unknown substance is polar. Blood is predominantly made of water, and water is polar. Sweet. We've just gone around in a circle over and over again, kind of repeating the same ideas. No problem. You're good. You can use this approach and this understanding as you go into your lecture test. So let's go ahead and look at this. If our unknown substance then is dropped into oil, which is a fat, right, which is non-polar, am I going to get my unknown substance dissolving in oil? No. No, because unknown is polar, oil is non-polar. This has to be like dissolving like. That's it. Boom. You just finished the first experiment, which is called solubility. Awesome. In class, you would do this with a beaker. It would be fun, blah, blah, blah. Sadly, we are not in class, but happily, we will not get COVID-19 staring at each other across the screen. So I want to be in lab with all of you, but I don't want COVID-19. So, we got to kind of take what we can get. All right. And literally, this is what we would do. Wait, what? Jensen's like, wait, So, no, I don't want COVID-19. Not sure about you. We're going on a no for COVID track. Exactly. I'm with you. Still, I don't want to go in. I'm just, what was the last two answers? You're so sweet. All right, so the last two answers were this. Actually, you tell me very quickly. If we know that the unknown substance is polar, will that polar substance dissolve in blood? Yes or no? Jensen, tell me what you think. Yes, you're good to go. Now, last question, same idea. If the unknown substance is polar, which we've established with this experimental part, will it dissolve in oil, which is nonpolar? No, you got it. All right, guys. Awesome, sweet. I love it. Good job. Okay, so that's no problem. So that's what we see in question 16. And remember, guys, just as a reminder, I am not banishing you from the headphones, right? If you want to hop on and give me an answer, no problem. The text is just kind of easy. You can chill out, But I don't want you guys to think that you cannot get on the headphone. You can. Just pop on and get off. All right, awesome. Good job, guys. That's it. You are now finished our first of three experiments that have to do with pH and also polarity. So the first one had to do with solubility, which has to do with polarity, right? All right, that's great. So now we are going to do our next experiment, which is still on page 16, and it is called pH and buffers. So you guys should be little experts on this by now. So we talked about this in lecture, but some of you may not have me for lecture, so let's go over some basics here. pH means what? Who can remember what pH means? Melissa's Friday lab under my groups must be removed. Okay, I'll go check it out. On my thing, I'm not sure what's there. Let me see. Melissa's Friday lab, office hours. Okay, I don't have another Friday lab on my Blackboard. So just know that the only thing there is office hours and this lab. Okay, so I've got a Tuesday lab, a Friday lab, and I've got office hours. Those will not be removed. They are part of the hours that are available. So as a reminder, guys, I have 4 classes up there, right? I have a Tuesday lab, which says Tuesdays. I have a Friday lab, which says Fridays. And then I have office hours, Thursdays and Fridays. Okay, guys? Now, I'm looking at my Blackboard really quickly here. I have one screen on my Blackboard, and those are my classes. I cannot remove those classes. So if you're coming to my class, you have to come to the class that says Friday lab at 5:30 to 8:20. I have nothing else that says my name except for office hours. Okay, guys? I totally understand, Portia, but I have nothing else on my Blackboard. My Blackboard has 4 links. Okay, if you want to, Portia, email me with a screen grab, because on my Blackboard, I have 4 links. I have a Tuesday lab, I have a Friday lab, I have 2 office hours. Those links cannot be removed. So if there's something you see, you need to email me individually, and then we'll deal with it. Right now, there's nothing on there that I see other than that what cannot be removed. Okay, no problem at all. I have no problem with that, but you have to email me because I cannot change anything that's currently there, and I don't know what extra links are there. So it may be an IT issue as well. So just email me individually because 47 people made it to the correct class. Okay, so I don't know what you guys see. I only know what I see on mine, and there's nothing else but one Friday lab. So email me. All right, so let's go ahead and look at this. So pH is what, guys? It stands with what? pH is what? Again, just email me if you see a link that I don't have. All right, potential hydrogen. Exactly, right? So potential hydrogen is the amount of hydrogen in solution, right, guys? So if I have a lot of hydrogen in solution, what am I? Acidic or basic? It's acidic. Very good. Now, if I have less hydrogen in solution, what am I? If I have less hydrogen in solution, I'm basic. We often see this basic scenario with something called hydroxide, which is OH negative. All right, guys, does that make sense? So in here, we often see that OH negative, which means, oh, I've taken that hydrogen out of solution. All right, guys? So now what do I see here? What I want to know is, am I more acidic or am I more basic, right? And we are looking at at the idea of maintaining a pH that is conducive with blood at 7.35 to 7.45. All right, guys? So this is my homeostatic goal for blood, right, specifically. Now, the stomach looks different, right, because the acidic content in the stomach is going to be much lower in order for digestion, which we learn in A&P2. So this is the basic of pH, right? Because you can have an acidic pH, right, a lower pH, or you can have a more basic pH, which is going to be a higher pH. And they're both related to the amount of hydrogen ions in solution. Okay, so that's what we're looking at here. And again, here you see a couple of redundant pictures, which have to do with neutral, 7. More acidic going to the left, which is a lower pH, and then more alkaline or basic going to the right. So often I write 14, but sometimes you'll see that this pH just kind of peters out at 12, depending on how a particular image is made. So that's what we're looking at here, potential hydrogen. Now, this is a big deal here because, again, what does this show us here? This shows us the amount, the pH that is a homeostatic value for blood. We want to maintain our blood at this pH. And then again, notice how 6 to 7, right? And that's kind of low, actually. You're dead, right? We don't go very high and very low in terms of blood pH before you die. So we get into some really problematic scenarios when we get too acidic acidosis or too basic alkalosis. Okay, guys? So that's what we're looking at there. So we're going to actually look at what happens if we have a buffer. So the idea of this next experiment, which is on page 16, has to do with pH and buffers, right? What we just talked about were pH. Now, how do we maintain this range of optimal functioning, 7.35 to 7.45? Well, as the experiment title implies, it's a buffer system, right? So that's going to be a buffer. So in this case, you need to know this. So you need to know this for your lecture. And this is going to be, yeah, exactly. Buffers are critical. So what you need to do here is you need to know this. First of all, if you don't know that carbonic acid is H2CO3, you need to know that by today, right? Because you need to know it for lecture test. You need to know that this HCO3 is bicarbonate. So you need to know both the molecular formula here, H2CO3 is carbonic acid. You need to know the name also. And you need to know HCO3 negative is bicarbonate. So you should be familiar with both of those and be able to easily kind of manipulate those terms. And then hydrogen, of course, is hydrogen. And that's what we're looking at when we're talking about the idea of being acidic or basic, right? We're looking at the amount of hydrogen in solution. And that's the definition, right? Potential hydrogen. So in this particular case, we want to have a buffer which has a weak acid, carbonic acid, and a weak base, bicarbonate. When you have these two together, we maintain a particular pH. And the definition of buffer is resisting the change in pH. Okay, so that's what we see there. We're resisting the change in pH. We do that with this buffer system, the carbonic acid bicarbonate buffer system. So that's our whole thing here. That's what we're going to be kind of showing with this particular experiment. So the way we do that is, and again, guys, if you didn't write this down or take a screen grab or something of this, because this is really, really important, and it's really important for lecture as well. But this is our buffer system. This is what we're looking at when we're trying to either become a little bit more basic or a little bit more acidic, depending on where we are in our pH or how far we are from homeostasis. But in this particular experiment, we are not actually looking at being outside of homeostasis. It's just that's how we apply this in the blood. All we're looking at is, hey, do I have a buffer or do I not have a buffer? And again, if you watch the video, no problem. If I want you to watch something in preparation for lab, I'll send you an email. So watching the videos, that's great. It's excellent, but we're going to go through each of the steps here. So again, in the next slide, if your numbers don't match up with this because you watched the video, guess what? Totally okay. The idea here to see a pattern within this experiment. Not that we need to see identical numbers, because you won't see identical numbers even if you had the lab, right? You'll see different numbers with different groups in the lab setting. So the idea is that we're finding this pattern to show us, well, which of these unknown solutions is going to have the buffer. And that's really what we're looking at here. So this particular chart that you have is going to replicate the one that you see on page 17. I watched the video and wow, so sorry. I'm so sorry. Well, Terilyn, let's go through this, and I hope that it becomes very clear. And it's always hard when you go in with a with a resource and you come out more confused. We try to avoid that. So let's see how we do today. Let me know if it clarifies things. I hope it does. So just to where we are, the chart you see in front of you is not going to have the same numbers, but again, that's okay. What you see is that there is a result that would be the same as you see on the video. And my chart here looks just like the chart on page 17, only I took out the last column, which is concentration of hydrogen ions in solution. Okay, we'll talk about it, but again, I'm focusing on the main idea here, which is pH needs to be maintained in the body. How do we maintain that pH? We do it by using a buffer. Okay, and we talked about this in lab in terms of how do we use that buffer system, right? We're going to donate hydrogen if it's too basic. We're going to take hydrogen out of solution if it's too acidic. That's kind of the basic idea here. So let's run through this experiment very quickly, guys. So we're going to look at the initial conditions. Initial conditions means what we start with. We haven't done anything yet, right? We have two beakers. We have beaker A, we have beaker B. Notice that they both measure at the same pH. Again, these numbers are going to be different than the lab video. That's completely fine. The idea is that the results, they align in terms of what pattern we should see. So beaker A, beaker B. At beaker A, beaker B, we're going to take our initial pH reading, and both of them here measure the same. That's great. We don't know what's in beaker A or beaker B. Well, your lab coordinator would, because they would set this up for you. But we don't know. We just know that there's a beaker A and beaker B, and we've measured them with our pH meter, which you would use in lab, which we don't use here. And we're going to see what happens as we go to next step, right? So we've gone from our initial condition to step one, which means we're going to start to manipulate what we have here, okay? Well, we're going to start manipulating it ourselves. Beaker A and beaker B obviously are not the same contents, but we don't know the difference between them. All right, guys, so step one, beaker A, and step two, and step one, beaker B, we are going to add 1 milliliter of hydrochloric acid. Now, if I add hydrochloric acid just generally, what would you expect to happen to that solution? You expect it to become more acidic or more basic. It's a hydrochloric acid. So I can dissociate hydrogen and it will go into solution. Exactly. So it's going to become more acidic. We would assume, right? That's our assumption based on how this would work. Now, what I want you to notice is that my initial condition is 7 pH for both of them. Step one, beaker A, what happens to beaker A when I add 1 milliliter of hydrochloric acid? What happened here? Careful. Here is beaker A, and here is beaker A. It becomes slightly more acidic, or the pH decreases. Careful with that, guys, because that's absolutely true. What happens is people confuse themselves with these terminologies. So it becomes slightly more acidic is the exact same thing as saying pH decreases. Okay, guys, so just make sure you're familiar. So being more acidic is the same thing. I know, I'm with you. My glasses are insane. So run and grab them if you need them. So becoming more acidic is the exact same as decreasing the pH. So just make sure you're careful with that so that you don't misinterpret a question because you're rushing on the test. So here we go. Beaker A with 1 milliliter of hydrochloric acid becomes 1 pH. It decreases 1 pH. It becomes more acidic. Now let's look at beaker B. Beaker B then, and I'll kind of help you out here so that you can see what I'm doing, because it is a little bit hard to follow. So beaker B has initial condition of 7 pH, and after I add 1 milliliter of hydrochloric acid, what happened? It remained the same, right? Now, pH should not remain the same when you add an acid unless it has what? It has a buffer. Boom, you're done. So we're not quite done. We want to see if that's actually the case. We're going to go to step two, and we're going to actually double the amount of hydrochloric acid that we add, right? So I'm going to add 2 milliliters of hydrochloric acid. Now, normally, with no buffer, what do you expect to happen if you add more hydrochloric acid to a solution? What do you expect to happen if it has no buffer? It should become even more acidic. So we're going to see what happens here. Beaker A, from step 1 to step 2, what happens to beaker A? It becomes what? It goes from 6 pH to 5 pH, which means what? It's more acidic, right? Okay, great. We've now gone from 7 to 6 to 5 pH. Now, let's look at beaker B here, and we're going to look here. happen there when we add another 2 milliliters? It stays the same. Just to highlight this point, beaker B started at 7. With 1 milliliter of hydrochloric acid, it remains 7. With an additional 2 milliliters of hydrochloric acid, it remains at 7. Which means what? Which one of these beakers has the buffer in the solution? A or B? B. We're done. We now showed by adding an acid component which one of these beakers has the buffer inside. Tara Lynn, does that make sense? I'm only picking on you because you made a comment. Does that make a little bit more sense? The second column. Oh, yeah, the second column. The second column basically, oh, by the way, guys, the second column is described on page 16. It has to do with a general scientific concept called scientific notation. It basically means that if you have something like this, 10 to the, sorry, that's not right. If you have something like this, 1 times 10 to the negative 4, you're going to go, if you have something like this, 1 times 10 to the, this is a very quick scientific notation. If you have 10 to the negative 4 and the pH would be 4, you're going to go to the left 4 times. Exactly. So if you start here with your 0, with your decimal point, and you go 4 times, 1, 2, 3, 4, I end up with 0.0001. So I started my decimal point here, and then I moved to the left because it's negative 4 times. 1, 2, oh wait, 1, 2, did I do it wrong? 1, 2, 3, 4. No, I did it right. Thank you, Halima. I did it wrong or right? Halima's like, yes. Right, thank you. All right, yes, the top is right. But it basically just means that when you have something like this, you're going to take the number and you're going to start your decimal point. You're going to go to the left, negative 4. You're going to go to the left. Yeah, Tara Lynn, I overthink this. I actually hate scientific notation. I shouldn't. I have a biology degree, but I really don't like it. Yeah, that's all there is to it, literally. So if you want to practice it, like if you're totally into it, simply go ahead and review page 16. And it literally will work. It'll take you through each of those. I know, it's horrible. I mean, maybe I shouldn't admit that, but that's the way it is. So that's it. That's it. So on page 19, the answer to number 2, what is the concentration of hydrogen in a solution with a pH of 4? The pH of 4, and what's confusing about that question is it actually looks like this. 1 times 10 to the negative 4. That's what they're asking about. That means a pH of 4. The change is based on the initial condition. Tanya, are you talking about the experiment or are you talking about scientific notation? Clarify for me if you mean the change here in pH or are you talking about the scientific notation? The number that we go to the left with. So 5 would be, yeah, exactly. Yeah, Gloria, exactly. You're just going to go to the left 5. Now, what's confusing here is that pH of 4, that's what it looks like first. So that's why we go to the left 4, because it's actually 10 to the negative 4. That's why. If it was 10 to the negative 5, Gloria, your example would be correct. The loss of hydrogen. The loss of hydrogen. The loss of hydrogen. The loss of hydrogen. Okay, so just generally, Tanya, and explain to me if I'm answering your question. The loss of, experiment 7.5. Page, give me a page number. It's okay, give me a page number, Tanya, really quick. The pH from 7 to 5. Oh, got it, sorry. Okay, so basically what happened in a nutshell is this. In order to find out which one of these has a buffer, right? Because this is the whole thing about the experiment. It's all about, well, how do we resist change in pH? That's what a buffer is. We're going to give you 2 beakers. 2 of them, we don't know what's in them, right? The initial condition is that we give you 2 beakers and they're unknown. So the initial condition is unknown. Because of the experiment, one of them, based on the experiment, has a buffer. One of them doesn't. So the question is, which one has the buffer? And we know the one that has the buffer because it resists change in pH as we continue to add hydrochloric acid. So A doesn't change at all. From A, we add hydrochloric acid, it doesn't change. A, we add, did I just mess that up? Hold on. What did I do here? Did I say A? See how bad I am? B. That makes more sense. Thanks, Jennifer. So B is 7. First hydrochloric acid addition stays the same. Second hydrochloric acid stays the same. And that's probably, sorry, I might have messed you up there. So beaker A changes when I add the first hydrochloric acid, and in the second addition of the hydrochloric acid, it also changes. That is correct, this way that I did it there. Tanya, does that make sense? Yeah, sorry, Jennifer. And for everyone paying attention, I hope I didn't mess you up. All right, awesome. So this diagram that I just drew with arrows, that's exactly what we're talking about. Yeah, you know. All right, Tanya, does that make sense?
Yes, that made sense. I figured I'd just get on the microphone instead of typing.
Go for it.
I was just asking on the side in the book, it was talking about the concentration of hydrogen ions in the solution.
So the concentration of hydrogen ions in solution is explained on page 16, and that was the whole thing that everyone was talking about with the scientific notation. It basically means, it comes from something called Avogadro's theorem, or equation, and it's a way that we can describe the concentration of something very, 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 very small, right? Because hydrogen is just one ion, right? It's ion. So in order to think about something that's very, very small in concentration, we use this idea of 1 times 10 to some sort of subscript, right? Which is the actual pH. So I wrote this before, 1 times 10 to the -4 as an example, because that's the example we have in on the blue box on page 19. This basically tells us how much hydrogen we have in solution. And so the way that you get this concentration is you basically use what's called the scientific notation. You take this -4 and you go from here and you go to the left. Negative means to the left, positive means to the right. If you're going to the left, it means that you don't have a lot of concentration, right? Because you're getting smaller and smaller. So in this case, if I go 1, 2, 3, 4, period, the answer becomes I have 0.0001 moles. There's a big M over here. Mole is simply the unit of measurement that we use for the amount of hydrogen in solution because it's so small. Like I can't use milliliters. I can't use something like that unit. I have to use moles because it's so small. So that's where the idea of concentration comes in. And it's how to do that is described in more detail under pH on page 16. So I don't have that column here because I just wanted to stick with the main focus of the experiment, where's the buffer. And then you can see the concentration by filling in the amount. Now, if you want to know how to do that, this here, how you write down this pH, it looks like this. 1 times 10 to the -7. That's a pH of 7. 1 times 10 to the -6, that's a pH of 6. 1 times 10 to the -5, that's a pH of 5. And then you take your 7 and you go 7 decimal points. You move your decimal point to the left 7 times. All right, sweet. That was a lot, I know. And I left it out primarily because it's just a very kind of straightforward mathematical moving the decimals to the left, and that's explained on page 16. I don't know that we ask on the lecture test, and I definitely don't ask on the lab test. Does that make sense, guys? So that's the concentration. You just move it over to the left. Yeah, that's it. And again, if you're like, what is she talking about? Just literally go over on page 16 and do a couple of those examples, and you'll get it. Okay, awesome. So that's basically it. That's what we did there. And that finishes the idea of the which one has a buffer. Now, it's a good idea to read the buffer information here because they talk about, well, actually, the buffer information is going for our next experiment. So again, good idea to read this because it kind of gives you, well, this is an experiment and this is what we would see. And this helps you understand the buffer system called the carbonic acid bicarbonate buffer system. All right, awesome. That's it. Now we're going to do page 18. Although the prerequisite information is on page 17. In other words, what's a buffer exactly, right? And I talked about the buffer with the first slide before we got into the first experiment. Now, there are 3 experiments in this exercise. Again, we would do this in lab, so you can see kind of the way I'm doing it. I'm just giving you the important pieces, and then we're going to look at what happens each step of the way. So the big thing about the buffer for this last one is we want to maintain this pH, 7.35 to 7.45, right? And with that 7.35 to 7.45 range, we have to be able to manipulate the pH by either making it more basic, right, going this way if it's too acidic, or conversely, we want to make it more acidic if it's too basic. So we talked about this in lecture, so we should be familiar with this. And so what we're going to do is we're going to show that in this experiment, okay? So you got to know your carbonic acid, this. You got to know your bicarbonate, which is this, right? And you got to understand what makes something acidic, lots of hydrogen, and what makes something basic, less hydrogen, okay? So that's kind of where we're going with this. Now, this is your page 18 in a nutshell, okay? So the question then is, we've got a beaker. It's a small beaker, and it has water. There's nothing else. It's just water. So in the experiment, you take a straw, and you blow in it. So you blow, 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 blow. Now, if you're blowing into the solution, what are you actually blowing out? So we know we breathe in oxygen, and what do we breathe out when we breathe? Carbon dioxide, right? So we're going to actually show how this works in terms of a buffer if I change the pH out of what the buffer will do if I add more, in this case, I make it more acidic. Okay, exactly. So here, this is the equation, right? I actually really like this slide because this kind of sums it up, right? And this equation sums up what's happening in the experiment. Okay, so what we have here is carbon dioxide. That's your, what would be called the initial condition, right? I have water, here's my water, and then I blow in it. So I'm going to blow carbon dioxide, right? And then here I've got the carbonic acid, right? Now, where did I get this carbonic acid from? Well, I'm basically playing chemistry puzzle. What do I mean by that? It means I simply rearranged my carbon, my oxygen, here it is, 1 carbon, 2 oxygens, right? And then I have 2 more, I've got 2 hydrogens, which go here, and then I have 1 more oxygen, which goes there. So I haven't lost any of these atoms. I simply rearranged them, right? So I've gone from the carbon dioxide with the water. I've rearranged my molecules, and I now have this intermediate form, which is very highly reactive. So this carbonic acid is not really stable in this form, but that's the first form that we get, right? Now, if I'm carbonic acid, right, remember that I have this extra hydrogen here. So as I quickly change, as I quickly undergo this reaction, what happens with my hydrogen and my carbons and my oxygens? What happens here? What's my next step that happens pretty quickly? What do you see here? What happens to the hydrogen? So I'm going to get 1 hydrogen leaving, right? And then what molecule do I end up getting if I get 1 hydrogen leaving? What do I get from that? I get bicarbonate, exactly, right? Now remember, if I add enough of this, if I add enough of these hydrogens, if I dissociate hydrogens into solution, what ends up happening here? I become acidic, right? So when I have lots of carbon dioxide in the blood, very quickly, because obviously it's in a water-filled environment, I'm going to have this chemical reaction take place because this is what it will do. I'm going to make this carbonic acid. Now, a carbonic acid is not acidic in and of itself. It's acidic because it has an extra hydrogen that dissociates or drops into solution. Because remember, nobody said that an acidic solution has a bunch of carbonic acid. No. A carbonic acid is an acid because it has an extra hydrogen that I can drop into solution, which is exactly what happens in this example. For every carbonic acid I have, I'm going to be able to dissociate 1 hydrogen and make this bicarbonate. And actually, next semester you're going to learn this bicarbonate is how we transport CO2 out of the body. Most of the CO2 does not dissolve in water, right? I need to put it into another form. So this is just something to think ahead with. But again, what makes this, what is it, guys, that makes this, what is it that makes something acidic? I have to add more what to solution? Just the hydrogen. It's just the hydrogen. So again, the reason why carbonic acid is acidic is because it has that extra hydrogen, right? And the reason why bicarbonate is a weak base is because it can pick up that hydrogen. And then we have this scenario where I have less hydrogen in solution. Okay, so that's what we see here. That's why the end result of this particular experiment, I first take a pH reading of my plain water, which is just about 7-ish. As I add more, as I blow carbon dioxide, I'm going to take a pH reading, and that pH is going to do what? As I blow more carbon dioxide into solution, what happens? Does it become more acidic or does it become more basic? The pH is going to lower or drop or decrease. It's going to become more acidic. And that's exactly what happens because the hydrogen concentration, this bad boy, is going to increase. Okay? And that's what you get in this particular case. So it's a great example of seeing the change in pH because you measure it after you blow carbon dioxide into the water. But it's also critical because next semester, that's what happens in how we move carbon dioxide from the blood out at the lungs. We're going to actually exhale and expel that carbon dioxide with exhalation, with the half of respiration. All right, guys, and that's it. That's your third experiment that has to do with pH buffer system solubility, and this one is just buffer. Any questions about that? We would do these all in the lab as the experiment that you saw in the video if you watched it. And again, you didn't have to. You know, that's okay. We did it here. All right. Any questions about that? So, yes. So what? So that's it. That shows you how a buffer system kind of works as you're looking at an unknown substance. No. So the equation overall interchangeable. Yeah, it is. It absolutely is, right? I can go in both directions. Do you guys see these arrows here? They mean it's reversible. I need an example. In what way? What example do you need? So if I have a pH of 10, yep. You won't have it in the blood because you'd be dead, but go ahead. In your example, you have 10. Yeah, you would be. But yeah, go for it. I mean, pH is pH in terms of moving regardless. And if you add H, if you add hydrogen and you have a pH of 10, you're going to go to the left, right? You're going to become less and less basic, more and more acidic. So if you add H, if I just add H, remember, just add H yourself on a piece of paper. It's going to become more and more acidic because our initial condition was for acidity, I have a lot of H. For basicity or alkalinity, I have less H. That's why we often see these hydroxide because we've taken the H out of solution. You got it? Sweet. All right, guys, any other questions? Remember, you can basically getting it back closer to 7. Yes, in this case, yes. Now, for blood, of course, the pH that we're looking for is, getting it close to 7. But really, precisely 7.35 to 7.45. Is that considered negative feedback? Yes, it is, Gloria. If my pH is too high, I got to bring it back down. If my pH is too low, I got to bring it back up. Negative is not bad. I mean, it just means that I'm going from to the opposite condition. If my temperature is too high, I got to go low. If my temperature is too low, I got to go high. That's negative feedback. Fingers. You help me more than you know. No. Sometimes I help, sometimes I annoy. It's a vacillating thing, right? Nobody's perfect. All right, guys. So, any questions about that? Didn't want to get off track, but is alkalosis making your internal issues too soluble? Okay, let's go back for a second. All right, guys, really quickly. Because I found this online somewhere. Some of these funky pictures I literally get from like Google Images. So I like this picture because it's cute, it's funny, and it talks about alkalosis, right? So if I have alkalosis, what it means is that I'm becoming more alkaline, right? As the name implies. Acidosis means I'm becoming more acidic. So what happens with alkalosis is, boom, I'm outside of 7.45. Notice that I'm not way outside of 7.45. I'm just outside of it, which is outside of homeostasis. So in that case, it's not so much what happens is, and I'm going to give you a very basic idea because we're going to talk about it in next semester. So the idea of acidity in the blood is this. if there's more acidity or less acidity, that impacts the binding of oxygen. So pay attention to this for a second if you care. If you don't, plug your ears, because this is not on an exam this semester. Okay, guys? This is a question that I'm answering, so feel free to plug your ears. If the reason why I need this function, this pH, is because, well, proteins function in that range, but also because of the affinity to oxygen. Now, this is way too much information than you need now, but basically, depending on if I'm acidic or basic, I'm going to either bind more strongly to oxygen or I'm going to release oxygen. Now, that makes a massive deal, because if I'm at the lungs, I want to bind my oxygen hemoglobin. Why? Because I want to get that oxygen in and send it to the body. Now, if I'm at the tissues, I don't want to have a strong bond with oxygen. I want to release that oxygen. So, and I'm not going to go into any more detail because you don't need to know, but basically the idea is this. That pH really, really matters to whether or not that oxygen binds tightly, which is what I want at the lungs, or whether or not that oxygen releases easily, which is what I want at tissues. So, Tanya, that's what I'm going to kind of give you today. Is that good enough for now? Because alkalosis and acidosis is going to mess up your bonding. And we're going to see that next semester. So, guys, everyone can start paying attention again. AMP2 builds on this. You have this stuff. By the time we add 4 more new buffer systems, you guys are going to be like knocking it out. And if you're going to be a nurse, all of this stuff is the basis of pathology. You can't understand how the body breaks down if you don't understand how it works properly. Homeostasis. Yeah, it's so cool. So this semester I like it, but AMP2 is where you kill it. So that's it. Okay, guys? Any other questions? Okay, that's what we're looking at. Good job. There's not, the lab discussion questions, again, you have a weak acid and a weak base. So if you're a weak acid, you donate the hydrogen, the carbonic acid. If you're a weak base, you accept the hydrogen. You are the bicarbonate. We've talked about this so many times now. And that's basically the idea there. Okay, so we're going to go to exercise 4. Good job. And I want to make sure that we can get to mitosis, because I'm going to go into a lot of detail there. All right, exercise 4. Now, right, guys, ready? So I want you guys to read the manual, because it's important and there's information. But what I want now is I want you to flip to page 31. Okay, guys? I want you guys to flip to page 31. I want you to look at the large cell model. Now, remember what I told you. Like 99% out of 70 questions, most of those questions are going to be, here's a picture, what is it? Okay? Now, on page 31, you will see under large cell model, it says, and I quote, you should know the structure, in other words, label this, and the function of the following. Now, it's simply for the function, you need to know what the organelle does, right? So, because you need to know that for lecture, and all my lecture folks, I didn't cover this in lecture. I didn't. And I love you guys, but I don't feel bad, right? Why? Drop it in a flashcard, be done. Get your point, get a point here. Okay? That's like low level, this is what it does. The smooth endoplasmic reticulum, it makes fats. The rough endoplasmic reticulum, it makes proteins. The mitochondria makes the ATP, it has DNA. Right? That's what I'm looking for. That understanding of function will get you your points that you need on both lecture and lab. Okay, guys, does that make sense? So what we're going to do here, literally, is this is where you guys get to be more engaged. Okay, great. Melinda, I'm glad. So I'm literally just going to go top to bottom. Why? Because guess what this is? This is getting ready for the lab midterm. Okay, if you know all these with a function, you'll get your points. Now, guys, does spelling count? Does spelling count in this course? It does. So you gotta write it out. There's no way you can avoid writing it out, because we're kinesthetic people, right? Sitting there and staring at the name of something doesn't make sense. Should we use this for the upcoming test? For the functions, yes. But what I'm going to be telling you primarily here is for our lab. So for the upcoming lecture test, you want to know the functions. What I'm going to be going over is what you'll see on the lab. That's why you can see that I'm on page 31, getting ready for the lab model, getting ready for the lab midterm. So it's a good question. Is there a place to print this out? Yes. If you go online, guys, remember, no problem. If you guys go online, there's all the images, right? So you're going to go to lessons under the lab, of course, and you're going to scroll down. You have all the images there. So the image, actually, this one here is, I just got it from online because it was easier. I mean, if I can find something labeled, I'm going to get it labeled. But here is, oh, wrong way. Oh, what'd I do? Wait, hold on. Wait a minute, what just happened here? Oh, hold on, guys. My PowerPoint messed up. Sorry, give me a second. Oh, okay. I'm going to start with this, but I'm going to upload something else while we're talking. I have in my other PowerPoint, which I uploaded the wrong one. It's okay, I'll upload it. I have an image from Blackboard. So this image is from just the web, but in my other PowerPoint, which I'm going to upload while we're talking, I have an image from Blackboard. So I didn't notice that this was a little different. So let me go ahead and upload, and then we'll keep going. All right, guys, I don't want to spend a lot of time here, but I added the image to another PowerPoint that I want to show you. This should be it. All right, sorry guys, I don't want to spend time. I just want to show you the image so we can go back and forth with it. All right, anyway, I'm going to add in just a second. I don't want to waste time. In my other PowerPoint, I have the image from Blackboard. It looks exactly like this. It just doesn't have a black background. So this is the image that you will see, and you can see all these images on Blackboard. So you want to definitely go on Blackboard as you're going through this, find the large cell model, print it out, and then label it. And then print one out that's not labeled, and then just write it out. Sorry, guys. No problem, Jennifer. All right, so let's start here, and then I'll see if I can get a chance to kind of pull up. Because in my other PowerPoint, I have an unlabeled one, and I like that because then I can go back and forth. But I don't want to waste a lot of time. So basically, we're going to go from top to bottom, and I just want to kind of show you where we are. So number one, nucleus. As you can imagine, the nucleus is all this inside here, right? Remember, this is a model. So we're going to see some of this stuff that's oversized. So this is going to be the nucleus, just like you would see in a slide, right? The nucleus is going to be the very inside, darker stained in a slide. So I'm going to give you that information at the same time. So when we get to the mitosis slides. So the nucleolus are here, and I've got this little ball here, right? So depending on what source you look at, there's like 1 or 2 nucleolus, right? We often just say that there's more than one nucleolus, nucleoli. You can see it's just the ball in the middle. Okay, now this is labeled as well, so that's hoorah, that's great. Okay, so the chromatin, which we'll talk about later when we talk about mitosis, is fourth, so I'll come to that in a second. So what we have, and if you're wondering what I'm doing, I'm literally going from the top of this list to the bottom because I want you to show you what it is. You're going to print out a copy of this, preferably in color, right? So if you don't want to print it out, you can just make a digital version, right? Just stick it in a Word document and test yourself from that. Still label it so that you can look at one unlabeled and one labeled. Okay, guys? So then the next one on page 31 under large cell model, you should follow me here, is going to be the nuclear membrane with pores. So here is the nucleus, right? So the nuclear membrane, also known as the nuclear envelope, but stick with the terminology we have here, right, guys? So don't give me nuclear envelope and say, it's the same thing. Okay, but I want the labeling as per the book. So nuclear membrane with pores, that's the blue part here, or gray, and it has little holes in there. Now, my other students couldn't see it when I enlarged it. Apparently from your end it looks wonky, so I have to keep it this size. All right, so that's the nuclear membrane with pores. Awesome. The chromatin is this kind of brown stuff inside. In the lab, it looks like that's that you use to clean super rough pots. And so this is what's called chromatin, okay, or chromatin. And we'll talk about that when we talk about mitosis. So that's what you want to be able to label. Now, this arrow, there you go. Guys, if you want to use the pointer, you can. So go ahead and do the pointer. That's fine if you want to. Just notice with this picture, the circle is not what they're pointing at. They're pointing at this rough stuff over here that kind of looks like hair all around there. That's the chromatin. We're going to talk about that in a little bit, okay? Here, we're just literally going over this, just like we did with the microscope, right? Boom, 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 boom. And then the next thing is the rough endoplasmic reticulum, okay? The rough endoplasmic reticulum here is, it's got, you can see little bits of red on it, right? It's blue with little bits of red. I can see it here. I can see it here, right, guys? You can see it here. So it's that blue part with a little bit of red, okay? Now, when you get this when you get this image without the black background off of Blackboard, you're going to be able to see this because you're going to be able to expand it by just simply enlarging it, right? You just cut and paste it, put it in a Word document. Or you can cut and paste it and put it in a PowerPoint if that works better for you. You can create a whole slide deck around this and go through it and test yourself. Okay? All right. So that's what we have for the next part. We did the rough endoplasmic reticulum. Now, smooth endoplasmic reticulum is not going to have those red dots on there, right? It's going to be the blue like this, right? The blue like this here, where there is no dot on it. Now, this is a little bit small, so I'm sorry about that, but when I expanded, my other class said it was very hard to see. So that's what we have here. We've got the smooth endoplasmic reticulum, called smooth because it's the endoplasmic reticulum, but it has no ribosomes, right? So in the rough endoplasmic reticulum, what are we going to be making? So here's my rough endoplasmic reticulum, guys. It's got the red dots. They just look like red, the color red. What am I going to make on the rough endoplasmic reticulum? Because those red are ribosomes, exactly, proteins. Now, what am I going to make on the smooth endoplasmic reticulum that does not have these ribosomes on it? What do I make on the smooth endoplasmic reticulum? Fats, exactly. That's right. So I make the fats in the smooth endoplasmic reticulum. Awesome. That's it. Awesome. Now, free ribosomes are just like the ribosomes that are attached to the rough ER, but they're free, right? They're not attached to anything. And that's what you have here. They just look like these red little dots there. And I've got them over here also. So free ribosomes for the cytosol. Yeah, they're just floating around the cytosol. They're not attached to any other organelle, right? So ribosomes are considered ribosomal organelles, right? But the ribosomes on the rough ER are attached to the rough ER. That protein that's made gets removed from the cell, where the free ribosomes, those proteins stay in the cell, and they have various functions. Samantha, did I answer your question for the cytosol? Okay. All right, awesome. And then we're going to keep going in mitochondria. Mitochondria, they look like little Cheetos, the Cheeto Puffs. So that's it there. That's the mitochondria, Cheeto Puffs, right? In other things, they look like beans, but here they do. They look like Cheeto Puffs. Hey, if you can look at that and give me mitochondria if we ask you for it, sweet. Again, spelling counts. That's why you got to write it out, right? There's just no way to avoid writing it out. So the mitochondria is where we make the bulk of our ATP. 38 ATP gets made there. And the mitochondria has its own DNA. So it's called mitochondrial DNA. You get that from your mom. And that's how your siblings all have the same mom, right? Now, as for dad, it's a different story. Anyway, so that's mitochondria. And it's really, really important, right? And we can make more mitochondria because you have DNA. Okay. All right. So that's what we have. Centrosome. Centrosome tricks people because we have so many C words very quickly, it becomes kind of confusing. So a centrosome is going to be part of, let me just make sure I got this. Yes. So this one here is labeled correctly. That's how we want you to label it. It looks like two little Christmas candies. It does. Looks like two little Christmas candies. So those two little things they're actually kind of like they're more almost like a protein type of it's almost like a micro tubule, but they're fat. So here, this is one centrosome. So each of these two centrioles make up one centrosome. That's why we ask you to write centrosome /centriole. That's what you have to write to get the correct answer. Why? Because that's what we want. It's on the list. It's spelled there. That's what we want. For your information, one centrosome is going to have two centrioles. And again, they look like little Christmas candies. The little Christmas mints. All right, guys? Now, this confuses people every semester, but you should be able to find it in the video. Lysosome is the purple balls, and that's correct, right? That's how we do it, right? The lysosomes are these big purple balls, okay? And lysosomes they bud off of the Golgi apparatus, which is very cool. They literally come off of the Golgi apparatus, and they have the proteins, the enzymes, which are proteins, inside of them that catalyzes reactions. In other words, your bacteria and stuff like that breaks them down. That works. I like it. See, I never even thought of that. Like literally, L large, P petite. Never. Didn't even cross my mind. Some things are in my mind and some things are not. So that's perfect. Excellent. Lysosome large, peroxisome petite. I love it. I'm going to steal that from you, and I will not mention your name. I'm kidding. It's like plagiarizing, which is not funny, but all right, awesome guys, good job. So I love it. See guys, so you guys have ways of thinking of things that I only have my own particular perspective, but all of you have something else. Yeah, isn't it? It's a great one. I love it. So that's perfect, right? So lysosome is going to be large. Just stole it from you. Peroxisome is going to be petite. The yellow is going to be the peroxisome. Peroxisome is involved in metabolism, specifically breaking down the fats. So you'll often find that in like places like liver, which is an important metabolic organ. Okay, so things like that. So you need to know those functions for lecture, and you need to know them for lab. So guess what? Knock them out now. Get your points on lecture. So if you start studying this late, you want to knock it out now so you can get those points for lecture as well. And then you know it for your lab, which is great. It's a twofer. All right, awesome. Vesicle. A vesicle basically is a sac. Now, you can't call it a sac because that's not the word we want. We want a vesicle. And basically, we have exocytosis and endocytosis, or bulk transport using these vesicles. And they're just right here, right? That's actually showing something that's either probably leaving the cell. But we can enter the cell in the same idea, okay? And so here we go. They're just like this and like this and like this. So those are going to be the vesicles. So what happens is we kind of shove these things all full of proteins, and then we just send them out, okay? A lysosome can kind of fuse with something else and destroy things. Like it can fuse with a bacteria and then just break it down and destroy it. All right, so we're almost done with that. Plasma membrane. Now here they call it the cell membrane, which is fine, but I don't want that. I want the plasma membrane. Why? Because that's what's on page 31. And that's it. Like we're not arguing, right? We're gonna look at the list, or we're gonna learn it the way the list says, because that's what the list says, right? We don't have to worry about, well, the internet says this and this and this. It is a cell membrane, and we often call it the cell membrane because that's what it is. But we want you to know it as the plasma membrane, know it as both. Here we would just simply put an arrow, and we would say, well, we would try to put it as close to right there as possible. And we would just say, what is this? And you would tell me it's the plasma membrane. That's it. Okay. The plasma membrane, it surrounds the cell. It keeps all the organelles inside, and it allows cell transport. Boom. There you go. And then again, peroxisome, that's the next one. And we've got them as petite, the yellow balls. Lastly, cytosol, cytoplasm. You know cytosol is the water. Cytoplasm is kind of like a gelatinous type of substance that the organelles kind of are sitting in, if you will. And you can't really see it as a 3D substance. It's just the yellowish part that everything's sitting on, right? So be careful. If we point at the yellow part, we're not pointing directly at an organelle. That's cytosol and cytoplasm. All right, guys? So in a little bit, this was super boring because if I had the other slide, I'd go back and forth. In a little bit, when we take a break, I'm going to go get that slide, and we're going to kind of just quickly you guys are going to go over it a little bit with me. All right, guys. So guess what? We just went over the large cell model. If you know all of those, and you know the function, right, the basic function, and you can spell it, you will get your point on the lab midterm. You will be asked about these on the lecture, so you might as well knock it out and get a twofer, right? You're going to be asked about some of these organelles. Now, how many? I don't know. Maybe 2. Which ones? I don't know. I don't make the test, right? So that's why we're prepping you for the test. Every class gets an equivalent test. Not the same test, right? But they get an equivalent test. And none of the instructors know what's on it. So I got to prep you for all of it. All right, guys. Any question about that? That's the large cell model. Nope. So that makes sense, guys, right? You understand this is an example of what you would see on the test. This cell model, it's actually the exact same cell, but it's just not on a black background. That's on Blackboard. Get in there, print it out, just label it, and get your points. Right, guys? So remember, guys, everybody, stop what you're doing and listen to Miss Nikolic. Please, get anything we cover that week, you must knock it out by the end of the week. You guys are Friday, so I'd say Tuesday, Wednesday latest. Everything we cover that is underneath getting ready, that's found under getting ready for the lab midterm, knock it out by next week. Because that way you're super on top of the material. You get to apply it to your lecture, so you get a 2 for points, right? And you're able to stay on top of it as the material starts coming in fast. Does that make sense, guys? That's how you think about prepping for this lab midterm. All right, guys, make sense? It is straightforward. Get those images off the blackboard. We'll test you off those images, right? All right, awesome. Great. That's it. Sweet. Okay, so let's keep going. And we're on exercise 5. So what are we supposed to do today? We're supposed to do 3 things. We're supposed to do exercise 3, which we've done, exercise 4, which we've done, and exercise 5. So this is a great time for me to give you guys a 5-minute break. I'll go quickly rummage through my file folder and find the one with the empty